Today's talk is by Dr. Christopher Reid, and it's my great pleasure to introduce him to you today. Dr. Reid is the Associate Professor of Science and Technology at Bryant University. He's originally from Ontario, Canada, and Dr. Reid received his Bachelor's of Science from Laurentian University, his Master's of Science from the University of Waterloo, and his PhD from the University of Guelph, all in Ontario, Canada. Before joining the faculty at Bryant, Dr. Reid worked at the National Research Council in Ottawa, where he developed mass spectrometry tools for glycobiology, specifically in the analysis of lipid-linked oligosaccharides of N-linked protein glycosylation biosynthetic pathways. The resulting methodology was an enabling technology for glycoengineering efforts in bacteria. Hope that's okay, Chris. I did that right.
Yeah, you did a good job.
Thanks. Dr. Reid's work spans the fields of chemistry and microbiology and has taken a number of different paths over the years, including the development of novel antibiotics, for which he's got a putative patent for, I believe, in the works. And as a member of the RICA community, Dr. Reid has begun exploring the world of marine microbes, which he will talk a little bit about today. So please join me in welcoming Dr. Chris Reid, and please maybe be a little extra supportive as his beloved Montreal Canadiens find themselves in a hole 2-0 against the Tampa Bay Lightning. And so he might be feeling a bit despondent this morning. So welcome, Chris, and all yours.
Thanks, James. At least they played well. Didn't appear in the outcome last night. Okay, so thanks for having me. So if you're a bit of a movie fan, this title is a little bit of a spoof off of Dr. Strangelove, right? Black and white satire from the 1960s. Okay, so I'm going to tell you a little bit story about how I went from doing chemistry and antibiotic discovery to how I'm now looking at carbohydrates in marine dinoflagellates. Right. Before we get going, just today's July 1st. It's Canada Day back home, so happy Canada Day. Right. And before I get really rolling into this story, got to highlight the team. So this summer we are a pretty full group. We got 5 undergraduates in the lab, Nick, Jet, Kate, Paige, and Elimelech, who's not in the photo at the moment, as well as 2 full-time lab research associates, Mika and Bryant. Right, so today's work is going to mostly follow the story currently of what Nick in the lab is working on this summer and what Caitlin had started during the spring semester this past year, and a couple previous students as well. All right, so what we do in the lab, right, we're kind of a hodgepodge of a bunch of different things under kind of the moniker of carbohydrate chemistry or glyco work. So, right, what we do in part is glycochemistry, right? So we do, you'll find in our lab a bunch of organic synthesis, purification and analytical analysis of biopolymers. Right, but that kind of doesn't really cover everything we do, right? We also do glycobiology, right? So we are interested in carbohydrates and their function in microbes and organisms. So you'll also find students in our lab doing everything from basic microbiology to actually gene expression studies, right? So the better term is probably glycoscience to explain what we do, which is we're interested in the biology and the chemistry of carbohydrates and their role in biological systems. All right, so when I was back in grad school, my grandmother, when I would come home for the summer, was like, "Oh, you're studying carbohydrates, kind of like what's in your diet?" In grad school, and I like to highlight the awesome hair there, right? But I would say yes, but it's so much more. All right. So how did I go from biomedical related research, antibiotic discovery, vaccine development to marine science? Well, way back in 2010 now, I joined the faculty at Bryant University. at some point in my first couple of years, I actually attended a faculty workshop at the Metcalf Institute. I believe it was on writing op-ed pieces, right, for newspapers, right? And at that workshop at the Metcalf Institute, I ran into, and actually we were in the same group working on a breakout assignment. And that's where I met Suzanne Mendendour over at the Graduate School of Oceanography. Nothing came out of this right away as a collaboration. That's where we first met. And a few years later, right, we started talking again about stuff. And this is how I started becoming interested in investigating the biology of dinoflagellates. And in particular, what we're going to be talking about today is the dinoflagellate Oxyrus marina. All right, so before we get to the carbohydrates, this story actually starts somewhere a little bit different, and actually begins with talking about lipids in this organism, right? And this is what I'm going to talk about first here is work done by a former postdoc in my lab, Brad Hobrich, former research technician and undergraduate student, Kiana Rouhani, and a former undergrad, Kai Lu, who did a lot of this work. Right, so working with Suzanne, they had been studying the dinoflagellate Oxyrus marina and looking at what happens to it when it undergoes long-term starvation, so lacking a food source in its marine environment. And Oxyrus marina is a very widely distributed marine dinoflagellate, and it is kind of used as a lab proxy for studying these organisms. Right, so Suzanne came to us and said, we've been looking at Oxyrus marina under starvation conditions, and what they had observed in their lab is that as Oxyrus marina undergoes starvation over a long period of time, upwards of 21 days, what they saw was this decrease in lipid content in the cell. And this lipid content, and it was this decrease in lipid content was proportional between, whoops, my bad. My laser pointing here, right? She was looking at two classes of lipids. First is polar lipids, so a little bit of intro biochem here. Polar lipids are found in cellular membranes and they have a charged hedge group. And what I'm showing here as an example is very common polar lipid found in membranes, which is phosphatidylcholine. And then we have our neutral lipids in the cell, and these are mostly energy storage, right? So think as in fat vesicles, and these are referred to as triacyl or diacyl glycerols. They do not have a charged head group and are composed of 3 fatty acids linked via esters to a glycerol, right? And over the course of observing the starvation period, right, Suzanne's group had noticed that proportion of polar lipids and the neutral lipids, so think membrane lipids are our polar lipids, our neutral lipids are more of these storage lipids, triacylglycerols, were decreasing over time as the cell volume was decreasing, but they were decreasing proportionally, right? And that ratio of total polar to neutral lipids in their experiments was looking appeared to be the constant at all times. It was decreasing over time, but the ratio between the two classes of lipids remained constant. Right, so this is where we kind of joined in and started collaborating. And what we wanted to look is, so when we look a little bit closer at this phenomena, does this ratio of the polar and the neutral lipids in Oxyrus marina actually are constant? All right, so some of the research questions that come out of this, right? So kind of a big umbrella question here is, what kind of changes occur, and particularly what we're interested in our lab in particular are molecular changes. So what changes to biopolymers, biomolecules like lipids and proteins and carbohydrates occur in dinoflagellates like Oxyrus marina when they are deprived of their food source? That's a really big question that one can go in a very large number of ways, and it's kind of hard to pin down an answer. So we kind of got to come up with a little bit tighter questions to ask here, right? So from that, okay, we want to know what changes occur, but from Suzanne's data, we saw these differences in neutral and polar lipids, and another question we can ask is, why do polar and neutral lipids decrease in this proportional manner in Oxyrus marina, right? So that's a little bit more narrower. And then finally, a question that we can really kind of tackle right away to start getting at these higher order questions is, what changes are occurring to that neutral lipid fraction in Oxyrus marina? And the reason why we chose the neutral lipid fraction is some work in Suzanne's lab had suggested that there were differences that were in that neutral lipid class in this organism. All right, so the first thing we did, and this is work by a lot of hard work by Kai Lu, who graduated a little in 2020, and Kiana in the lab that left in about 2018, right? So what we do in the lab, we obtain Oxyrus marina cells from Suzanne's lab. We would then subject them to organic solvent extraction to extract all the lipid content from those cells. And what we did initially is, right, from Suzanne's data, they were doing flow cytometry and just looking using dyes, total classes of molecules. We want to kind of take a little bit closer look and kind of look at the polar and the different classes of neutral lipids present in the cell as that starvation progressed. So the first technique that we use in the lab, it made for relatively quick analysis, is using something called high performance liquid chromatography, which I'm showing an instrument here. And if you're not familiar with it, it's basically a method for separating mixtures, right? So we have two major components in HPLC. Is we have a stationary phase, or an HPLC column shown here, which interacts with our mixture. And we have a mobile phase, or HPL solvent, and we play off of the different the equilibrium interaction between molecules interacting with the HPLC column stationary phase and being soluble in our solvent. And as the lipid mixture is applied to that column, we change the solvent system over time, and we can separate a very complex mixture of lipids into its different classes. And so what we're able to do using HPLC is basically separate total lipids in the cell into total polar lipids. MAGs and DAGs are a class of neutral lipids. So MAG stands for monoacylglycerol, DAG stands for diacylglycerol, and DAGs stand here for triacylglycerin sterols is the component. So think the red and the green bars in this graph are the breaking apart our neutral lipid fraction a little bit further. Sterols are things like cholesterol, right, and steroid like molecules in the cell, right? So when we looked at different time points during starvation, what we noticed is that the ratio of the polar to the neutral lipids remained pretty much constant throughout that starvation. The cell volume decreased, but the proportion of polar to neutral lipid stayed the same. So suggests something is occurring as starvation to maintain homeostasis within that cell. We need to have that balance between these two classes of lipids needs to occur, right? So this gave us a little bit more information on what was going on, but we needed to dig a little bit deeper to get at what was happening. Right, so we then use another type of chromatography. So what we're mostly on this project up to this point, our contribution was more of an analytical chemistry standpoint in separation and analysis of the biomolecules. So to get a little bit deeper and get a little bit more information about that neutral lipid class, right? Kiana, Brad, and Kai Lu did a lot of gas chromatography. So this is very similar in terms of how it works compared to HPLC. But instead of having a liquid solvent flowing over as our mobile phase, what we use is a carrier gas, typically helium, and we inject our sample in. It gets put into the gas, heated up, and placed into the gas phase. It then travels and interacts with a column that's upwards of 30 meters long. That's getting pushed through that column via that carrier gas, and then we detect get the separated components on the other end. And what we get is a chromatogram that looks something like this on the bottom left, right? A series of peaks. And our gas chromatography is equipped with a mass spectrometer, so allow us to identify the the component that is under each of the peaks in our chromatogram. And what we noticed is as O. marina starved, what we saw is a huge increase in the number of fatty alcohols, right? So fatty alcohols are related to fatty acids that are in lipids, are actually derived from them, right? They are long-chain alcohols, right? Usually fatty alcohols, depending on the definition, are organic alcohols with carbon chains of about 5 carbons or longer, right? So when we looked at Oxyrhis marina in the active fed, we noticed a huge range of fatty alcohols present, right? And what we noticed, and we also compared this to its food source, which it feeds on photosynthetic algae, right? We could be able to see all the fatty alcohols that were derived from its food source. And we also noticed that Oxyrhis marina actually takes its food source and actually does what's referred to as trophic upgrading. What we saw is fatty alcohols that were also observed in its food source. Also, Oxyrhis marina took those and made them longer, right? So by extending them longer means there's more energy packed in these molecules, right? And what we noticed in Oxyrhis marina is there's a high tendency for what's referred to as unsaturated fatty alcohols. Same type of thing as unsaturated fatty acids, right? Where we have the presence of a carbon-carbon double bond, one or more in that molecule, right? So this is pretty interesting. It took us a while to kind of sort through this and kind of figure, okay, well, why is it producing these fatty alcohols, right? And what seems to be occurring, and how does this really tie into the fact that that polar and neutral lipid ratio is remaining constant as that cell is starving and shrinking in volume, right? Well, it's because as it's starving, it's starting to go through what's referred to as autophagy. It's starting to eat itself to extract nutrients, right? So what's happening is in Oxyrhis marina, right, we have our polar lipids here shown here on the left, and we have our neutral lipids, right? Both sources of fatty acids. So fatty acid component is what I'm highlighting here, and we have the same thing over here in the triacylglycerol, right? The cell will hydrolyze off fatty acids, right? These can then undergo enzymatic reaction and be reduced to fatty alcohols, right? So they're pulling these fatty acids off of the polar lipids and the neutral lipids at the same proportion. Some of them are getting converted to fatty alcohols, and then they are being stored as a high energy food source as wax esters, right? And as Oxyrhis marina is starving, what we noticed in our gas chromatography analysis is a reduction as starvation proceeds to a reduction of these wax esters, because it was using those preferentially as a food source in starvation. And wax esters, think of these get used as an energy source to produce ATP in the cell. All right, so that's kind of how we got into looking at dinoflagellates. And we had, looking at this and okay, there's these changes in the neutral lipids, and being a carbohydrate chemist, we sat there and went, okay, well, if the lipid profiles are changing, I'm wondering if there's, to bring it back home, what changes are occurring in the carbohydrate fraction of the cell. Right, so that brings us to the second part of our story, right? What about the carbohydrates, right? So carbohydrates have a myriad or a ton of biological roles, right? Carbohydrates that can be found attached to proteins as glycoproteins, as shown here. We also have glycolipids, right? That tend to be, I'm showing here stuff on the cell surface, right? So we have glycoproteins where we have carbohydrate groups covalently attached to a protein. Same thing with glycolipids where we have a variety of carbohydrate structures that can be bound to a lipid and embedded in the membrane. And actually, this is like a month old, is the newest form of what's referred to as glycoconjugates, right? Carbohydrates attached to some other biopolymer is glycoRNA, right? Which is found in on the cell surface of several eukaryotic cells. And this new discovery, maybe at least no more than a month or two old. Right, so some of those roles of carbohydrates in cells, right? So they form the backbone of nucleic acids like DNA and RNA. That they're kind of the forgotten carbohydrate polymer, right? They are very important as cell surface receptors, right? So think an immunology standpoint, right? Interaction between, actually, if we go into the pandemics, think of the vaccine production, right? When we get infected with something, right, we will present molecules on our surface. Some of those molecules are actually going to be glycosylated, right, with a carbohydrate structure, and that is can be part of what's recognized by our immune system that allows us to raise antibodies. Cell surface receptors are also important in cell recognition, so adhesion between and communication between two cells, right? Important helping protein folding. It's known to be involved in cell-cell communication, so two cells talking to each other. And this is kind of what we're going to be, where our line of research is kind of taken, is with the carbohydrates and oxygen is maybe they're using it as a cell-cell communication tool, right? So think presenting different carbohydrates on the surface can communicate to other cells in the vicinity that changes into environment or status of a given cell, right? Glycosylation in cells is also important, where proteins get localized in a given cell, right? And if we're talking bacteria or fungi or plants, carbohydrates can also have a structural role as well. And very important in immune signaling. A lot of, there are quite a few immune disorders that are rooted in aberrant glycosylation. Right, so we're not studying in a biomedical standpoint. We are looking at marine dinoflagellates, and looking through the literature, not a lot is actually known about dinoflagellates and their glycosylation machinery and pathways. What's mostly known is in a few dinoflagellates that are symbionts with marine coral. Right, so so what we're going to look at here, and what we're looking at moving forward in the lab, is changes to protein glycosylation, right? So if I go back a slide here, we're interested in carbohydrates that are attached to proteins sitting on the cell surface. All right, so in dinoflagellates and then across almost all eukaryotic cells, right, protein glycosylation occurs in a similar way, particularly when we're talking about N-linked protein glycosylation, which is where our carbohydrate is linked to a nitrogen on an asparagine amino acid on that protein. So protein glycosylation and is initiated in the endoplasmic reticulum in eukaryotic cells, where we build up a large structure composed of N-acetylglucosamines, which are these little blue squares here, and mannose sugars, and this is referred to as a high mannose structure. Right, this is attached onto a protein of interest, or a protein as is being synthesized off of the ribosome, right, in that ER. Once that protein is glycosylated, it is then exported and trafficked over to the Golgi body in cells, right? And the Golgi body is where we get all the diversity in our glycosylation patterns in eukaryotic cells. We have enzymes, so think it's actually a very energy expensive process. We put on these really, right, you see the base structure for every carbohydrate starts with 2 N-acetylglucosamines and 8 mannose residues, right? And then they traffic it to the Golgi, and there's enzymes that cleave away some of those sugars that were already put on and then puts on new ones, changes the linkages between those sugars, and coming up with 3 general classes, right? So your high mannose structures, which are basically, we don't name them anything exciting. We name them what they look like. High mannose has got a lot of mannose sugars on it, right? Hybrid is kind of the between our high mannose and complex, right? And all complex and hybrid are as we start having additional sugars, but other than N-acetylglucosamines and mannoses in our structure. And this is where we get branching, diversification, the structure, elongation, truncating, right? So what we decided to look at in the cell after finishing off with the lipid is we said, okay, well, if we got these changes in lipid and starvation that are occurring, what is happening on the cell surface in terms of that glycosylation pattern to the protein, the glycoproteins, right? And is there a change that's occurring that's maybe communicating this lack of food source to other organisms in its environment? Right. So how we go about studying this is we take our Oxyrus marina cells, right, which are on its surface have glycoproteins with a variety of different carbohydrates. I'm just showing one here because they take a while to draw, right? And we treat those cells with an enzyme called PNGase, which is stands for protein N glycosylase, right? And it cleaves specifically only carbohydrates that are linked via nitrogen to a protein. Right, so we're getting a subset of all the glycoproteins present, right? And it cleaves them off and it releases them into solution, right? So one of the problems in carbohydrate chemistry is they can be very hard to analyze. They don't absorb light very well. They are hard to analyze by mass spectrometry unless you do some sort of chemical derivatization to them, right? And they are very diverse, so it can be very hard to analyze by other techniques. So to try and make our life a little bit easier for analysis is what we do is we take these released carbohydrates from the surface of Oxyrus marina, and we do a little bit of chemistry to them. And what we do is, so every single carbohydrate that gets released by our enzyme PNGase, right, has what's referred to as a reducing end. It has a aldehyde functional group that is present, right? We can exploit that because everything else in the carbohydrate structure is nothing but alcohols. And we run a reaction that's referred to as a reductive amination. We take our carbohydrates mixture, we throw in a molecule that's referred to as 2AB, which stands for 2-aminobenzamide. And what happens is we place it in there in the presence of a reducing agent. And what we end up with is a derivative of our carbohydrates in which we have linked that 2AB group onto the end of every carbohydrate. And what this does is it makes it easier for our carbohydrates to be analyzed by mass spectrometry. So one of the problems with carbohydrates is that not super easy sometimes to get them to adopt charge in a mass spectrometer to allow us to observe them. This 2AB glycan now, when we derivatize it, allows us to more easily place a charge on that molecule and be able to analyze it by things like mass spectrometry. It also gives us a chromophore, something that absorbs light. And this 2AB group, when it's covalently linked to the carbohydrate, actually fluoresces. So it gives us a way to track our isolation purification of our samples. Right, so how we go about analyzing these mixtures? Well, we don't get much material, so it's hard to separate them out into all their individual components and analyze them. So what we do is we take those mixtures of our 2AB derived glycans, and we use something referred to as multi-mass spectrometry. And that MALDI stands for matrix-assisted laser desorption ionization. And basically what we do is we take our carbohydrate mixtures, mix it with what we something called a matrix, which helps to absorb light from a laser beam, right? So lasers make everything cooler and funner, right? So in MALDI mass spectrometry, we use a laser beam, strike our sample that's on a stainless steel plate. That causes the molecules that are on that plate to be ejected from that plate, right? And what happens is we get transfer of a hydrogen, right, onto our molecules to impart a positive charge. And then that goes through to the mass analyzer. And what we do in a MALDI, basically think the lighter molecules they're all accelerated with the same amount of energy. Lighter molecules will have a higher velocity. They will pass faster through the detector and they'll be detected first. The heavier ones will come later, right? Those go through the mass analyzer. And what we get out of this is a complex spectrum of a bunch of masses, right? So on the surface, masses don't tell us anything. What we then have to do is take all this data, and plunk it into some bioinformatic databases, such as one we use frequently is glycomod, which is a nice invention, as it allows us to input these masses for what could be carbohydrate molecules into this database, and it kicks us out possible carbohydrate structures that correspond to the given mass. Right, so before we start looking at the data we got out of this, right, just a refresher on the three major types of protein end-linked glycans, right? We got that high mannose structures here, right, which contain nothing more than these blue squares are N-acetylglucosamine residues. These green circles are mannose sugars, right? Hybrid is, we'll go over to complex first. Complex are really very diverse in the types of sugars present in those types of N-glycans. We have things like sialic acids, which are these little diamonds, right? We can have other sugars added at different branching. Hybrid is kind of the mixture of, we get some of the features of the complex, where we have the diversity of different sugars in the glycan, still a fair number of that original high mannose structure present. All right, so when we went looking at Marina, we were really hoping that the glycans on the surface might be able to provide us some information on the nutritional status of this organism, right? So think Marina, right? So we have algae in the ocean, photosynthesizing, fixing carbon. Dinoflagellates like Marina will eat that algae, right, and trophically upgrade, basically increase the energy content of that food source, and higher orders of marine organisms in the food chain will feed off that. So think the status of kind of this low-level marine organism can kind of give us some interesting nutritional status on food webs in general, right? If there's a shortage of primary producers in the ocean, it could have detrimental effects to higher orders in a food chain, right? So first thing we decided to go looking at is, what does the surface carbohydrates look like on this organism when it's happy and fed, right? So fat and happy, what does it look like, right? So what we noticed is in Marina, when it's actively fed, right, we have predominantly two of the three classes of carbohydrates present on its surface, right? We have the hybrid structures, which have a large number of mannose sugars and some decoration with other monosaccharides, and a proportion of complex carbohydrates that get really decorated up. So I'm not going to show, we have dozens of structures, and this work has been done, right, was initiated over a year and a half ago by Abigail Enck, who has now graduated. Nick is now working on this now, has done a lot of work on going through the data we have and assigning structures, and Caitlin, who worked as a work study in the spring on this project. All right, so in the actively fed Marina, we see this solely hybrid and complex carbohydrate structures, and we look at the field of all the structures that we observe in our mass spectrum. We see a high degree of sulfation of these carbohydrates, so SO4 on there. So think we're a large degree of negative charge. So think of it, sulfate is there introducing negative charge into that molecule. We also see a high degree of fucosylation, right, which is common in fucosylation occurs frequently in all eukarya as a kind of a branching sugar at various points in end-linked glycans. Right. So we started with active fed, we got that baseline. Now we wanted to go see what's happening as we subject Oxyurus marina to starvation. So our first time point is looking at a 7-day starvation time point. And what we see when we look at O marina after 7 days of no food is we see a shift to purely complex and glycan structures, right? So lots of sialic acid. We see an increase in sialic acid. We see an increase in monosaccharides incorporated such as pentoses, right? We also see a shortening of those glycans. But overall, what we know is we still see sulfation, just like in the active fat, but we see now an increase in negative charge due to sialic acids are a sugar that have a negative charge group on them, right? So we see this increase in negative charge, right? So biologically speaking, what sialic acids can be, one of the many things they can be involved in is if I have proteins in the membrane that need to come together and aggregate to form a pore, right? If they are glycosylated, if I put a bunch of sialic acid sugars on my N glycans on those proteins, I'm imparting a lot of negative charge, is going to make it hard for them to come together and aggregate to form that pore, right? So there's a fair number of biological outcomes that arise from this increase in sialic acid, right? So our next time point in studying, right, is we did 7 days and now we're going out to 21 days starvation. And what we see here, we see a wide variety of carbohydrate structures again, but we don't see any of the hybrid structures. We really only see complex N glycan structures, right? One interesting thing to note is that we saw, we see at 21 days, we see a reduction in fucosylation, right? This particular sugar. We also still see a big increase in sialation at 21 days and sulfation. This is kind of interesting because making sialic acid in the cell is an energy expensive process, right? You would think something that's starving and lacking nutrients might not want to do something that's very energy taxing, right? But it seems to be doing that. And so we have this, again, increase at 21 days starvation, this increase in negative charge present on the surface. All right, so what does this data all mean? We're still kind of flushing through this and this is research kind of still in progress as we're trying to wrap up and kind of bring our data and kind of make some sense of it biologically, right? One interesting thing is that the cell surface end-linked carbohydrates on that surface seem to be able to give us some information on the nutritional status of the organism, which is kind of interesting because if we can have a surface marker on these cells and then potentially in other ecologically important dinoflagellates, we might be able to have a diagnostic marker that we can develop that could quickly assess the nutritional status of these important players in marine food webs, right? The glycobiology of dinoflagellates is really uncharacterized. So as a professor at a PUI, finding an area that's not occupied by a lot of people gives us a little niche area to study. And seems to be a lot of very interesting structures that are being produced. That sulfation, right, that I mentioned, we see here in our starvation with this increase in sulfation. Not much is known in dinoflagellates. So what we had to do is kind of go looking at other organisms. If we look at other organisms such as humans, when you see increases in sulfation and carbohydrate, it usually signals something not right with the cell, right? So in humans, increases in sulfation it can be associated with certain cancers, right? So we can think of that sulfation as maybe it's a signal saying that the cell is stressed, right? So the fact that the glycobiology of these organisms is really uncharacterized as a carbohydrate chemist, this is really quite interesting because it might give us an opportunity now to go in and characterize some potentially novel carbohydrate acting enzymes, in particular sialotransferases. So these enzymes that transfer these sialic acids onto the carbohydrates, so imparting those negative charges, as well as go and find maybe some of these sulfotransferases that are installing these sulfate groups onto the carbohydrates. Right, and this kind of leads us to a few of the areas where we're kind of moving into slowly as we are characterizing the glycans, right? And the first is, right, since there's so little known about protein glycosylation in dinoflagellates, right, we're thinking of applying our chemical biology tools and metabolic labeling strategies to maybe study these pathways in these organisms. And if the back orders can ever come in, Nick is going to be starting in the next week or two synthesis of sugar derivatives that are found in protein carbohydrates, right? So just showing an example here is what we are hoping to do is to take sugars like glucosamine here and install a chemical handle, right? This N3 group here is azide, right? Installing this group here allows us hopefully to be able to do some chemistry if it gets incorporated into the N-linked carbohydrates. So what we're going to be doing is taking the sugar glucosamine, derivatizing and installing this chemical handle here, this azide group, and then we need to somehow get this into the cell. So our first approach, and from the literature, one way to help and improve uptake of these sugars by cells is to throw a bunch of acetate groups around the sugars, and that allows the cells to take them up. So once we get this sugar made, our strategy moving forward is to get a test to see if Omarina might be able to take up our derivatized sugar with that azide handle to allow us to do some chemistry on it. And if it can take it up from its environment, we are hoping that it will be able to incorporate this derivative with our handle into the N-linked glycans on the surface, right? And that would mean we would have these azide groups, right, that will be unique in that cell to allow us to do some very specific chemistry, right? Not to go into that chemistry, but one thing we might hoping to be able to do in looking at that profile change is take dyes like fluorescent dyes that have their own handle on it, and we can do something that's referred to as click chemistry, right? We can take our labeled cells with our azide group, we can then some click chemistry, covalently click a dye onto our surface to do some fluorescent microscopy, flow cytometry, maybe help start to do a little bit more of the biochemistry and biology of carbohydrate synthesis in this organism. Right. Another avenue that we've started to pursue, and this is really in the ground stages, is looking at nanopores for glycan detection. And this is a collaboration with Jason Dwyer in the chem department at URI and his grad student, James Hagen, right? And it's our hope that we'd be able to use nanopores, which I'm showing here are basically these manufactured pores that are less than 100 nanometers in diameter, right? And we're hoping to use these as a method to detect neutral and charged N-linked glycans with these pores. And basically how it worked, we would have these surfaces with these nanopores in them. For solution on each side, we apply a current across that pore, which will cause electroosmotic flow across that membrane. And for neutral glycans, we can, hopefully the molecules as they pass through, how it works is we would get impedance of the current through that pore, and we get a change in that impedance over time, which we're hoping will give us some information on the carbohydrates that are present. We're hoping that this will give us not only be able to identify general classes of carbohydrates present in a mixture, but maybe give some structural information and information on charge status. Right. So at this point, we've had some success with some commercially available N-linked glycan standards, and we're hopefully in the next few months try it on some honest to goodness real live Oxyrus marina carbohydrate samples and see if we can identify the differences between active and starved cells. So that mostly wraps up this talk for today. I want to thank Suzanne for inviting me to work on this. It's actually been an exceedingly interesting project over the last several years. Her former technician in the lab, Amanda, fantastic, who left last year, but was a big part of the lipid work and the initial bit of the carbohydrate work. Robert, who's the undergraduate this summer in Susan's lab, who's been helping prepare all our fed and starved Oxyrus samples. To our new collaborators over in chem at URI, Jason and James, and have to thank Al Bach at the core facility at URI for access to the MALDI, which thankfully is now fixed and we will actually be using it tomorrow. Big thanks to the students that have worked on the lab. So Brad is a former postdoc from my lab who's now an assistant professor at Touro Medical School in Nevada. Abigail graduated a year ago, I think. Pandemic, I can never the time right anymore. Kiana was the longest serving member of my lab. She started as a high school student and then stayed on for a couple extra years as a research associate. And Nick, who is working on this project this summer as a CAIM surf student. And the rest of the Reid lab, we're a pretty big group. All right, we got Mika, Bryant, Caitlin, Lamelec, Paige, Jet, not all working on marine stuff. As many people here, there is as many projects in the lab on the go at the moment. And we also have a high school student that just started this week on our NSF grant, Monica Toma from South Kingstown High. And that pretty much wraps it up.
Great, thanks, Chris.
Over time.
Yeah, all right, I'll open the floor to questions before I jump in with mine. So any questions for Chris or Dr. Reid? Well, I'll start off, I guess. I've actually got two questions. One kind of big picture and one more nuanced, I guess, or one detailed. The big picture question. So these changes that you're seeing in terms of the surface chemistry, is it a response to a lack of nutrition in the sense of they can't do their normal metabolism, therefore the normal surface chemistry isn't available to them? Or is this a sort of, holy crap, I have to change what I'm doing and therefore, I'm not sure does that make sense, you know?
Yeah, no, I think it's, I have to change what I'm doing, right? So a lot of these carbs, particularly ones where we see starvation and we see all this negative charge building up, that's going to prevent multimeric complexes from forming. So it's probably preventing certain pores and transporters from coming together in that membrane because there's no food. It doesn't need to bring it through. Maybe they're energy transporter, so they don't want it coming together, expend more energy than it needs. But it is kind of interesting. Like it's expending a lot of energy to make some pretty complex sugars and to decorate these things while it's starving.
Right, but I imagine then that it must be, even though it's energetically costly to do that, it must be as a benefit. Otherwise, they wouldn't. Right,
exactly. Yeah, it's costly, but it has to be less costly than the alternative.
Right. Okay, and then my more detailed question is: you mentioned when you did the mass spec work, and you get these, you throw the information into a bioinformatics program, and it gives you potential carbohydrate structures. And I guess, how do you figure out from there? They give you 10, they give you 100, they give you, you know what I mean? What's
it all depends. So it'll go. So what you do is we take we input a table of all the masses that we observe, and it kind of goes through one by one. And it's nothing more than an arithmetic process where it's taking that mass and going, okay, what sugars can I put together to give me that mass? All right. So then we have to go through and go, okay, what do we know about? So there's very little known about dinoflagellate glycobiology, but the nice thing about eukarya is that basically the protein end-link glycosylation, the pathways are really well conserved across all eukaryotic cells. So what we then go and do, okay, what we already know from every other organism that's been studied is it has to start with two particular sugars back to back. So every structure has to have two N-acetylglucosamine residues minimum in its structure, because we also know that from the enzyme that we use to cleave them off the surface, if it doesn't have that at the end when it's linked to the protein, the enzyme won't recognize it and cleave it. So we actually have we know a little bit about like the fucosylation. If it's in certain places on the carbohydrate, our enzyme that cleaves the surface won't recognize and do the cleavage. So we actually, a little bit of what we know about the commercial enzyme and what we know from the rest of biology, we can go, okay, well, that's not a legitimate structure. What basically the database is doing is when I was in grad school, this didn't exist, and you literally sat with a calculator and a paper and pencil. Okay, I got a mass of 1,500. Okay, these two sugars plus these, and then basically just going through. And what this is doing is, like, in a matter of seconds, taking all those masses, doing that for you, and then you just have to go through and go, okay, well, that's not correct from what we know of biology. This is correct, right? Or if it picks one, it also gives you an error from what it calculates and what it observes. And we use that as a kind of a parsing tool to, but depending on the masses, in some cases we get 50 potential structures and it's looking for the right one.
And then do you just assume that if there are multiple possible, like truly possible structures, do you just assume they're in some proportion as
Yeah, so with that one, if they're all legitimate and they're all within the same error from what we see, right? Because the mass spectrometer is only giving us mass,
yeah,
we have to assume that all those are possible type of thing. So to dive deeper into that, I don't think we get enough material from our cell samples to be able to take those and kind of do really fine structural chemistry to determine what each of ours. So when we have that, we assume all of them are legitimate possibilities and probably are all part of that mass signal we see.
Okay, cool. Thanks.
No problem.
Go ahead, Jeff.
Hey, thank you, Chris, for the talk. Actually, I was just going to encourage everybody to, if you're in a place where you can and you're comfortable, turn your video on, so a little more of a discussion here. I was going to ask a question about the biophysics. So I'm kind of interested in that. Of course, when the composition of the membrane or cell wall is changing, you're going to be changing the underlying, mechanical environment, maybe the phase behavior of the membrane and so forth. Is that something that you look into as well, or have an interest in looking into?
I do have an interest. Yeah, that's definitely a route we can take on that lipid angle of that, is looking to see. We didn't do a deep dive into the polar membrane changes, right? So there's got to be stuff happening there, right? Which is going to have a whole bunch of stuff with permeability and all that stuff. It would be definitely very interesting to look at.
Yeah, that kind of stuff gets me excited. Being able to look at local viscosities and differences in lipid ordering, et cetera, that might prevent transporters from coming together. And
yeah, like I'm assuming there's gonna be changes in the lipid rafts and all that jazz.
Yeah. Maybe we'll have to follow up. I shot you an email too about a little follow-up meeting on another project we have. Maybe we can think of some collaboration in that area. That'd be
sure.
Cool. All right.
Yeah, no, I mean, when Kiana, I had just got rid of the lipid samples, like Kiana and Kailu in my lab. I had like 96 sample boxes loaded with, like, they were like machines of lipid extraction and analysis. Like basically they filled a freezer of samples for this lipid analysis. So
cool. Well, when we run our seminar, we always require that students have to, ask at least one question before we end the seminar. So I'll just leave you with that. Thanks, Chris.
Hey, no problem.
It's a good, I think it was another nice indication too from your talk from the beginning, talking about how you got into it through the Metcalf Institute. And then it's good for students to take note that the process of careers and sort of science itself, right, takes these weird little turns that you don't expect.
Yeah, like it just so happened Suzanne and I went to the same, like I figure this was like in 2012 or something like that. And then we ended up in the same breakout group. And then it was a couple of years after that, and then we got a stack proposal together, start looking at the lipid stuff. And it's just kind of been moving from there.
Well, I think it's important for the students to kind of attend these events and start to really see how scientists talk to each other and interact and really see that it's a very windy path that we take through the years. So,
well, it's funny when I go to CAIM events and it was like, oh, you're a lipid chemist. No, that's like a side gig.
All right. Any questions for Chris, Dr. Reid? I think we have a very quiet audience today. All right. Well, I guess if no one has any questions, then we will give Dr. Reid a virtual round of applause. And thank you, Chris, for that. And hope to see everybody next week at the next Brown Bag seminar. And